Hello, my dear girls and boys. A very warm good afternoon to all of you, and thank you very much for staying tuned to HashLearn. And as you can see on your screen is the today's topic. That is what we are going to discuss today. That is nitrogen metabolism. Okay. And before I start off the class, I hope you have enjoyed your puja vacation, and you are all fresh and refreshed and recreated to learn new things. So with the same zeal, let's begin the class. So now on your screen is the recap of previous class, what we have discussed in the previous class. So we were into the chapter mineral nutrition, where we have discussed the role of the different minerals, whether it's macro or micronutrient, and in the previous class we have discussed the deficiency of nutrients, mechanism of absorption of mineral elements, like how they are absorbed from the soil, then factors affecting the mineral absorption plus mineral translocation. And meanwhile, Joshna, hello, how are you? And I hope all is well with God's grace. Now on your screen will come the CPP question, the class practice pack question, where most of you have gone wrong. Okay, so here it comes. So the question was question number 4, and it's pretty easy. The deficiency symptoms of nitrogen and potassium are visible first in A, buds, B, senescent leaves, C, young leaves, D, roots. Okay. So most of you have opted for D, out of the league option actually. See, whenever we are talking about deficiency diseases of mineral nutrients in case of plants, okay. So two things you need to keep in mind. A is if it is a macronutrient or if it is a mobile nutrient, not micro. If it is mobile, that means it can be transported from one part of, or from the old leaves to the younger leaves, then that kind of a nutrients deficiency will be first seen in the senescent leaves. Okay. But if it is an immobile nutrient like that of a calcium, which is a part of the building block of the cells, like it's a part of middle lamella, so it is an immobile element, so the deficiency symptom will be first seen in the young leaves. Okay. So these two things which you are supposed to remember, and with reference to the nutrients. So nitrogen and potassium are very much mobile nutrients, and since they are mobile nutrients, their deficiency will be first observed in the senescent leaves, that means the leaves which are dying. Because I have explained this in the previous class also, that the young leaves which are there, okay, so that is the progeny, so that will be taken care of first. That is the priority of any organism, not only the plants. Okay, they will try keep and protect that particular part of the plants, and that is why the nutrients will move from old leaves to new leaves. But whenever the nutrient is immobile, then in that case you will see the deficiency symptom first in young leaves or birds. Okay, that will be the scenario, and that is why the solution is the B, that is deficiency symptom of nitrogen and potassium are visible first in senescent leaves. Okay, clear? And now on your screen will come today's goal. So what we are going to discuss today. So today our main aim will be on nitrogen metabolism, and what we are going to discuss in that is the nitrogen cycle, like how nitrogen is regulated in the like from our atmosphere to soil and back to the atmosphere, and then nitrogen fixation. So there are two types by which nitrogen or the atmospheric nitrogen can be fixed, that is physical and biological. And then we will be discussing about the mechanism of biological nitrogen fixation. So this will be dealt in detail, biological nitrogen fixation, and what will happen to the ammonia which is formed as a result of biological nitrogen fixation. So now let's start. So to start off with, we are into the nitrogen cycle. So in this, see, first let us know that when we talk about nitrogen, so when we say nitrogen, it is N only, and that is the symbol. And this is actually the dinitrogen or N2. But conveniently, when we speak nitrogen cycle or nitrogen this, nitrogen that, so here we are meaning the molecular form of nitrogen, that is the dinitrogen. Okay, so we are referring to that. Don't get confused that whenever I am saying nitrogen, it means it's an atom. No, it's a molecule of nitrogen. Further, this molecule of nitrogen is pretty inert kind of, that means it will not, it's not highly reactive. So that is the reason why it cannot be directly taken from the atmosphere, though the percentage of nitrogen in the atmosphere is almost around 78%. So that's the highest amount or the most abundant gaseous element present in the atmosphere, but still the plants cannot absorb it like that. And for us also, the source of nitrogen, okay, whether it's in terms of proteins or nucleic acid, the source is plant. So we are dependent on the plants, and plants in turn is dependent upon the natural sources of nitrogen, and from air it cannot take, and that is why it needs to be fixed, or it has to be combined with either oxygen or hydrogen, so that now that becomes absorbable by the roots of the plant. So in case of nitrogen fixation or in nitrogen cycle, there are a few things on which everything is dependent. First is nitrogen fixation, then you have the process of ammonification, nitrification and denitrification. So these are the 4 reactions which actually control the entire nitrogen cycle. So let's see what is nitrogen fixation first. So nitrogen fixation is the process of conversion of nitrogen into ammonia. So that means atmospheric nitrogen needs to be brought down to the soil, it needs to be fixed, and that is why it is called nitrogen fixation. And in this process what happens, the atmospheric nitrogen which is there, in that no hydrogen is there, hydrogen will be added and it is converted into ammonia. In turn, ammonification is actually the next process, or not in this, okay. It is the process of decomposition of organic nitrogen of plants and animals into ammonia. So in between what will happen, from ammonia we have the nitrification, because ammonia, as we have discussed like during nitrogen functions as a nutrient. So the the form in which nitrogen is maximally absorbed from the soil is the nitrates and not the ammonia, okay. So that is why this needs to be converted into nitrates. So for conversion into nitrates, you have, it needs to be converted first into the nitrites and then to nitrates, and this entire process of conversion of ammonia to nitrite or nitrate, that is called nitrification. So the ammonia so formed may volatilize and re-enter into the atmosphere because it is highly volatile, okay. So some of the ammonia may be converted into nitrate by soil bacteria. So inside the soil itself we have the bacteria which can convert this ammonia into the nitrates, and what will happen, then that will be taken by the plants, and this step is called as nitrification. The nitrate so formed can be easily absorbed by the plants and transported to the leaves. In leaves, nitrate is reduced back to ammonia to form the amine group of amino acids, okay. So that is the form only in which it will take up. Again, that needs to be converted into ammonia, but that is the story inside a plant. Now at the end what will happen, the process of reduction of nitrate present in the soil to nitrogen, it is carried out by bacteria like Pseudomonas or Theobacillus, okay. So these are actually denitrifying bacteria which will so in the soil we have the nitrates. So nitrates, in one hand it can be taken by the plant or it can be released into the atmosphere also. So if it is being released back to the atmosphere, that is denitrification. Nitrogen fixation brings it, you have here nitrification, and once it is released back into the atmosphere, that process will be called as denitrification. Now let's understand it with the help of a diagram. So you have the atmospheric nitrogen, and in this case this step is actually your nitrogen fixation, okay. So here, so nitrogen fixation is basically the process by which the atmospheric nitrogen will come down to the soil, okay. Any step which is bringing it to the soil, that will be called as nitrogen fixation, okay. Now once that nitrogen is fixed, so first form is the ammonia, and in case of biological nitrogen fixation or industrial nitrogen fixation, in nitrogen fixation we get the ammonia first, okay. Then from ammonia you will get the nitrite, NO2 minus, okay. This nitrite will then get converted into nitrate, okay. So here if we see this conversion of ammonia to nitrite is taken care by the bacteria that is called as Nitrosomonas. Okay, and then conversion of this is done by bacteria like Nitrobacter. Okay, so conversion of nitrite to nitrate is by bacteria Nitrobacter, and then it is this form in which it is taken up by the plants. By some plants it can be taken in this form also, but this is the form in which most of the plant or majority of the plants will uptake or take up the nitrogen. Now that nitrogen will be utilized by the plant and it will get converted into plant biomass. So that means it will become a part of plant protein or plant nucleic acid, then we have vitamins, or in chlorophyll. In all these places this nitrogen will be used. Okay, and then once the plant dies or the senescent leaves falls, so that forms the decaying biomass along with the animal biomass also, because some plants, because animals are directly or indirectly dependent on the plant. Okay, so from there that nitrogen is moving to animals also. So when these animals die or their excreta, whatever is there, that on that, if whatever nitrate is there or whichever nitrogen compound is there, that is formed back to ammonia, this step is known as ammonification. Okay, and after this again we will have the like here this can go from ammonification, you can have nitrite and nitrate, and this will take the nitrogen back into the atmosphere, that is called denitrification. For that we have the pseudomonas. Okay, so that's how the nitrogen keeps on moving or circulating in the ecosystem. So now we are moving to next. What is that figure, Joshna? 456 plus 4. So have you marked some answer? I didn't get you, dear. What exactly are you trying to say? Just or you are talking about some question. So now when we are talking about nitrogen fixation, we have two types of nitrogen fixation, that is physical nitrogen fixation and biological nitrogen fixation. So physical nitrogen fixation is once this nitrogen which is there in the atmosphere, it can also be fixed because of the thunderstorms or rain. So from this also they will take up the energy. Okay, so because of that you will have and that kind of a nitrogen fixation is known as physical nitrogen fixation. Okay, and in case of physical nitrogen fixation, the nitrogen which is present in the atmosphere, so that is simply the nitrogen. Okay, that is the nitrogen gas. Okay, so once there will be lightning or rain, so this lightning provides energy for the complete lightning, providing for this lightning will provide the energy for the fixation of the nitrogen. So you have nitrogen plus it needs to get combined with oxygen, and then the source will be either lightning or thunder, so that is providing the energy, and then you have this element or this compound is made, okay, NO, and then this will react further with oxygen to form NO2, and this is an oxidation process. Okay, this is an oxidation process, and now this will further react with water to form nitric acid or nitrous acid. Any of these can be formed. Okay, and in this form, that's why this comes down as an acid, and once it comes down on the earth, now this will get combined. Okay, So this HNO3 which is there, it will get combined, it can combine either with calcium or potassium to form calcium or potassium nitrate, and this is the form in which plants can take up the nitrogen. Okay. So this form, or if plants are getting nitrogen in this way, that is called physical nitrogen fixation. Now we are into biological nitrogen fixation. So reduction of nitrogen to ammonia by living organisms is called biological nitrogen fixation. So now we are into biological nitrogen fixation. So certain here, when we are talking about biological fixation, so since ages we have just read about rhizobium, but rhizobium is a symbiotic nitrogen fixing bacteria. Okay. Other than that also there are many bacteria which are free living in the soil, and they can also fix the atmospheric nitrogen. So say for example, when we are talking about this reduction of nitrogen to ammonia, so in this case we have the nitrogen fixing microbes, and they are classified into free living and symbiotic. Free living is also referred to as asymbiotic. Okay. So asymbiotic and free living are the same, and then this can be aerobic. If they need air for their or oxygen for their respiration, they are, so azotobacter is an example of aerobic free living nitrogen fixing bacteria. Anaerobic free living nitrogen fixing bacteria, there we have the Clostridium. Even in your books you have the example Rhodospirillum. Okay. So anaerobic free living. Then we have the cyanobacteria. Okay. Free living cyanobacteria is the Nostoc and Anabina. Okay. So these are the things where, so in this case what you need to remember is the name, and then whether it's free living or it is symbiotic. In free living also we have categories aerobic, anaerobic, and then cyanobacteria. Okay. So these are the examples, and then we have the symbiotic nitrogen fixation that happens if the symbiotic means the bacteria lives in close association with the plant. Okay. So it will get nutrients from the plant, in turn it will fix the nitrogen for the plant that the plant will utilize for its own metabolism. So this is kind of mutualistic relationship, and once it is symbiotic, so it can be, so the very common or classical example of symbiotic nitrogen fixation is found in case of leguminous plants, and the bacteria is Rhizobium. It can also happen in some non-leguminous plant, and bacteria there will be Frankia. Okay. So Rhizobium, and also this Rhizobium strains are dependent on the type of legume which you are growing. Okay. So that as of now we are not going to discuss the various types or strains of Rhizobium, but just remember Rhizobium as symbiotic nitrogen fixing bacteria. Now we are into the biological nitrogen fixation. So it needs three basic components. First is a reducing agent to transfer hydrogen atom to dinitrogen. Okay, because dinitrogen or nitrogen, N2 is the thing which we need to get converted into ammonia, and if it needs to get converted into ammonia, NH3, you need to add hydrogen into that. So you need an agent or the one which can give its hydrogen. Then it is highly energy dependent reaction, you need ATP molecules for that, and then we need enzyme system like nitrogenase. Okay, then molybdenum iron protein, leg hemoglobin. So molybdenum iron protein is actually part of enzyme nitrogenase only. You have a iron part, and then we have the molybdenum iron part. So this is the enzyme, and then you have leg hemoglobin. So leg hemoglobin is also termed as oxygen scavenger. Okay, because it will bind with the oxygen. Why? Because if the oxygen comes in contact with the nitrogenase enzyme, it will become inactive. Okay, so that is why nitrogenase requires a anaerobic condition, and to maintain that it is leg hemoglobin which will give it the environment. Okay, so it will eat away. Scavenger means it will eat away all the oxygen which is found in presence or which is there in the system. Okay, so leg hemoglobin, it is a pink color pigment similar to hemoglobin of vertebrates and functions as oxygen scavenger, whereas for us hemoglobin is very much required for transport of oxygen. Okay, we need oxygen, every cell needs, and there nitrogenase doesn't need oxygen, and that is why it will eat away the oxygen and protects the nitrogenase from oxygen. And the overall reaction which is there, it can be given by this equation, that is nitrogen, and there you need 8 electrons, 8, so you need a proton donor. This is required plus 16 ATP, because I have said that ATP will be required, and this ATP will be reduced to, or it will be broken down into ADP plus PI. This is inorganic phosphate, and as a result of this process we get ammonia, and hydrogen gas will also be released. So that this is actually the process of biological nitrogen fixation. So you got your answer, Joshka. Now we are into the nodule formation. So this biological nitrogen fixation, so we were talking about the free living and asymbiotic and symbiotic. So nodule formation is in the case of symbiotic bacteria, and specifically we are going to talk about the rhizobium. That means we are talking about the symbiotic association between rhizobium and the roots of leguminous plant. So root hair comes in contact with the rhizobium, okay, and it comes and the root hair will become curved like this, okay, and obviously the root hair also releases some chemicals and then bacteria. So that's a chemical interaction which actually brings the rhizobium towards it. The rhizobium is already there in the soil, okay, but only when these roots are there it will go and form a symbiotic association. Now plant forms an infection thread. So once it comes in contact with the root hair, okay, so bacteria will release some chemicals that will dissolve the plant cell wall, and a infection thread is formed, okay, and through that infection thread the bacteria will move inside, and it delivers the bacteria to the cortical tissue. So it will go till the cortex of the root hair cells, okay, and then bacterial cells divides and enlarge to form nodules. So once it reaches the cortex, okay, here it is the cortical zone, inner cortex and pericycle cells, here it will divide vigorously, okay. As a result of this, an enlarged protuberance is formed, and that is called nodule, okay. Nodules form contact with the vascular tissues to get food, that is why it has reached till the cortex, okay, and in turn formation of root nodules and nitrogen fixation occurs under the genes like NOD, NIF and FIX, okay. So that you need not to bother about, just remember till this that you have the root hair cells. Once the bacteria comes in contact with the root hair cells, it will get curved, okay, and then an infection thread is formed, and through this infection thread the bacteria gets delivered into the cortical zone, and then there it will divide vigorously, divide to form the nodule structure, and that is called root nodules, okay. Now this is what happens inside, or how basically this nitrogenase enzyme which is there, it will help in converting the atmospheric nitrogen into ammonia. So in this case, for the fixation of nitrogen, here what happens is you need, this is an electron carrier, okay. So in this case what will happen, see, first thing is you have the reduced peridoxin, it is in the reduced condition, that will get oxidized, okay. In turn it will, this is getting reduced, oxidized Fe protein was getting reduced, and now this reduced Fe protein, what it will do, it will transfer 2 electrons, and this will then convert the oxidized molybdenum iron protein. See, there are 2 subunits in the nitrogenase enzyme. You have the iron protein, and then we have the molybdenum iron protein. So these are two different. Okay. So where you have molybdenum, iron is there also, and even in case of in the free system also you have the iron. Okay. So free system means iron protein, there is one unit, and then you have molybdenum iron protein, that is another subunit of nitrogenase, and they play like pass the parcel. So electrons are being transferred, and as a result of this, finally once this gets reduced, molybdenum iron protein, this will further take up hydrogen ions, and then this will transfer this hydrogen, and now here we have the atmospheric nitrogen which we were talking about. This will get converted into NH. Then in second step it will again take another 2 ions, and then that will be NH2 only, and then that will be finally it will be converted into NH3. So let's understand it with a simple diagram which is there in your books also. So suppose this is the nitrogenase enzyme, okay, and this is the binding site for nitrogen. So these 2 nitrogen, so this is dinitrogen, 2 nitrogen with triple bond, okay. So now in first step what will happen? First step of reduction, okay. So that is why here you have the 3 things written, okay. In first round 2 hydrogen ions will come, okay. So as a result of this, what will happen? This will get, one bond will break and one bond is formed with the hydrogen, and you still have the enzyme, it is attached to the enzyme, okay. Then in next step 2 more hydrogen will be added, okay. So 2 more hydrogen are added. Then in the next step, that is the third step, so this is NH2 bonded by one bond, and then again 2 more hydrogens are added. So this will become, so this will break, and you have 1 nitrogen bonded with 3 hydrogen, that is the ammonia. So final product is the ammonia. So that's how the atmospheric nitrogen is fixed in case of the nitrogenase, or that is the biological nitrogen fixation, okay. And now on your screen is the previous class contest question. So what I have asked, it was mineral nutrients absorbed by roots move to leaves through A, xylem, B, phloem, C, sieve tube, D, companion cell. So in this the correct answer is A, mineral nutrients are absorbed by the root hair cells, and then they will follow apoplast or symplast pathway, and they are delivered into the xylem, and then along with the water column that gets transferred, okay. And anyways phloem, then you have the sieve tube and companion cell, that's component of phloem itself. So A is the answer and not B, C and D, and the first person and the only person to give the correct answer is the only person now also responding, and this is you only, Joshna. So a big round of applause for Joshna. Congratulations Joshna, you will get your free atom session credited by the end of this class, okay. And now the today's contest question will come on your screen. So Joshna, you can try today's question also, and you may win the atom session for today as well. So now on your screen will come the today's contest question. So here it comes. Thyroid disease of rice is caused due to deficiency of A, boron, B, iron, C, zinc, D, calcium. This is something I have discussed in my last class. And now answer the question. Thyroid disease of rice is caused due to deficiency of A, boron, B, iron, C, zinc and D, calcium. Now the question will go off your screen on my count of 5, 5, 4, 3, 2, 1 and next. Okay, so finally the answer has come from you. So in this now fate of ammonia, Okay, hello Apeksha. So Apeksha is also there and she has also attempted, and both of you are saying the same answer, so probably that could be the correct answer also. But now let's, so we are into the ammonia, which is what will happen to the fates of ammonia. So this ammonia was formed before, okay, just before the contest question we were discussing that the ammonia which gets formed as a result of the nitrogen fixation. Now we will have to see what will happen to this ammonia inside the plant, okay, inside the plant what will happen to this ammonia and how it will get converted and how the plant will utilize this ammonia, okay. But before we move into that, the one word which I forgot to introduce, that is bacteroid, bacteroid, okay. This is the structure formed when the bacterial or the rhizobial cells, okay, bacteroid, this is the structure, the rhizobial cells, when they divide inside the cortex of the leguminous plants, then there the structure, they will form a structure, a coagulated structure kind of, that is called bacteroid, okay. Outside to this bacteroid you have the peribacteroid membrane, okay. So peribacteroid membrane, okay, and then there you have the peribacteroid space, and then you around that only you will have the leg hemoglobin, so that it can eat away the oxygen, okay. Now we are into the fate of ammonia. What will happen to the ammonia? Like we have the fate of pyruvate when we talk about glycolysis, fates of pyruvate. Similarly we have fates of ammonia which is formed after biological fixation of nitrogen. So now this ammonia which is formed, that will be utilized to synthesize what? Synthesize amino acids, because the plant will need amino acids only, plus ammonia being toxic to the plant cannot have ammonia accumulated at one place, okay. So if you recall your previous classes of excretion, or if you have, or if you are in standard 12, so you know you have already read the chapter excretion. So there we have discussed ammonia is the most toxic nitrogenous waste, and it is toxic to the cells, and that is why once it is formed, okay, or once the cells take up, now that needs to be immediately converted into the usable forms which the plant can use, okay. So that is why that will be converted into amino acids, and the ammonia formed by nitrogen fixation is used for synthesis of amino acids. And there are two processes by which amino acids are synthesized. From where? From ammonia. The first one is reductive amination, okay. So in case of reductive amination, what will happen? The ammonia which is there, it will react with alpha ketogluteric acid, okay, and it will take that ammonia and it will form glutamic acid. So if we want to write it in the form of reaction, it will be ammonia will combine with alpha ketogluteric acid, and that will give rise to glutamic acid, fine, and it is catalyzed by enzyme glutamate dehydrogenase, okay. So that is the enzyme that will help in formation of the glutamic acid, okay. So that is one way by which it can be converted into amino acid. The other way is transamination, okay. So in case of transamination, amino group of one amino acid is transferred to keto group of keto acid. Now here glutamic acid is the main amino acid which transfers its amino acid. So generally it will be the glutamic acid which will transfer its amino group, that is this group, to form 7 other different kinds of amino acids, and the enzyme required here will be so why it will be, see here, because from glutamic acid, that is the first form acid, and it can easily give its amino group. So here if you see, the reaction will be it can go from any, I am just explaining the transaminase process. So, if we recall the structure of amino acid from the chapter biomolecule, so you have a carbon, then in that you have a R group attached, and then here we have the amino group, okay, and then COO minus, okay. So, this is the one group. This will combine with a keto acid or keto group of keto acid. So, you need a keto group here. So, this is the keto group, okay. So, now in this process what will happen, this amino will be transferred here and keto will be transferred to the first one, to the amino donor, and this process requires transaminase. The enzyme required here is the transaminase, and as a result of this what will happen, the R1 which is there, on that you will get the keto group and plus the other amino acid will be formed. So, this is the R2. So, amino group is transferred to that, and that is how the amino acids will be formed. And say for example, like aspartic acid which is there, that gets formed from where, if you oxaloacetic acid reacts with the glutamic acid. So, as a result of that aspartic acid will be formed, okay. So, this is one of the example. If we remove this, the same thing R1 and R2, and we take instead of R2 you take the oxaloacetic acid and other one will be glutamic acid, resultant what we will get, it will be aspartic acid, okay. So, that is how it happens, the process of transamination. Then there is one more thing, that is called amide formation. So, by replacement of hydroxyl group of amino acid by NH3 radical, okay. So, in this case what will happen, there is formation of amide, okay, like the synthesis of asparagine. See, asparagine and glutamine are amines formed from aspartic acid and glutamic acid in the presence of enzyme asparagine synthase and glutamic synthase. So, this happens when the hydroxyl group of amino acid gets replaced. So, this will be the case. See here what will happen, if I have glutamic acid. So, let us draw the structure for glutamic acid. So, this is glutamic acid, what I am trying to draw here, and then here we will have COOH, and this is your glutamic acid. Now, this if it combines with the ammonia will also require energy, and here glutamate enzyme will come, and as a result of this what will be formed, glutamine. So, what how this will be, the structure will be very same, it is towards the end. So, from here the hydroxyl group is removed. So, you will get glutamine, okay. So, CO and it will be NH2, and here we have the H. So, this is glutamine, okay. So, that is how, and then this glutamine if it reacts with aspartic acid, it will form the asparagine, okay, and there the enzyme asparagine synthase will be required. So, these are the ways by which the ammonia so formed will be utilized to form the amino acids required by the plant, and then it can be used in the protein synthesis, or as such it can also be used because we know nitrogen is a component of chlorophyll, it is a component of nucleic acid also. So, these are the ways by which this ammonia will be used, okay. So, ammonia needs to get converted into amino acid. I hope it is now clear, Joshna. Okay, fine, you have already responded. So, now we did this, we have come to the summary of the class. So, what we have discussed? We have discussed the nitrogen cycle. So, in nitrogen cycle, what are the things you need to remember? Just remember nitrogen fixation, then nitrification, that is conversion of ammonia to nitrite and then to nitrate again. Which bacteria are required? Like in first step you have nitrosomonas, in other you have nitrobacter. Then you need to remember what is denitrification, example of denitrifying bacteria. And then third thing, what is ammonification. That is all for nitrogen cycle. And then nitrogen fixation, there are two types, physical and biological. Biological is more important. In biological, you need to remember the free living and the symbiotic association. In free living, you have aerobic, anaerobic, then cyanobacteria also, an example of each of them. And then comes the mechanism of biological nitrogen fixation. So, how the root nodules are formed and how the enzyme nitrogenase will help in conversion of nitrogen to ammonia, and then how ammonia so formed will be utilized for formation of amino acids in case of plants. With this, we have come to an end of the class. So, thank you very much, Joshna. Thank you, Apeksha. I don't know who else were watching, but these two names I know very for sure. So, thank you very much for connecting to HashLearn. And now it is your CPP time, that is class practice pack time. So, now you will get your gift of CPP. So, don't miss to solve the question, because today's CPP question, you can see this was very simple question. Okay, but still most of you have gone wrong, right? So, that is why it is very, very you can take it as mandatory, you take it as my order, you take it in any ways, but please don't skip solving question. On this note, I take your leave. See you again in the next class, same time, same place, same face, but with a different topic. Till then, passport users start solving the question right away. Good luck.